In a few of my other lecture videos, I talk about some of the biological molecules like carbohydrates, fatty acids, amino acids, and nucleic acids. And with them, we go through their structure and some of the parts of the structure, which are functional groups. But I haven't really organized a specific lecture just on the functional groups themselves. So that's what this is. We're just looking at some of the most common functional groups found in the biological molecules. What are their characteristics and what is their structure? And that's really the main point of it. So I'll just give you a quick bit of background as far as, what is the role of these functional groups? Obviously, to add some sort of function. That function is largely going to be to add polarity. Right, to add some charge or partial charge to molecules. So if we think of the biological molecules in general, right, all of our biological molecules are carbon-based. Right? Carbon has 4 valence electrons that require pairing up with other electrons. So it forms 4 bonds. Those bonds are often with things like hydrogen, but they could also be with other carbons, and they're commonly with other carbons as well. Right, so I share an electron there, and then we get a couple more hydrogens in here. I'll put the carbon electrons. Okay, so each carbon now has its 8 electrons in the outer shell. The hydrogens all have 2, so they form covalent bonds with each other. These are called nonpolar covalent bonds because they share equally. And so what that means also, I forgot to draw that hydrogen, is that there's no charge. These hydrogens are not positively charged. There are no negative charges. There are no partial charges. It doesn't create the dipole moments because of the unequal sharing. The sharing is largely equal. So when this molecule, ethane, interacts with water, the water molecule is polar. The water molecule does not share equally between the oxygen and the hydrogen. So the hydrogen ends up feeling like it's lost an electron, being positive. The oxygen feels like it's gained electrons, being negative, and that's what allows water to interact with other polar things. This molecule isn't polar. So if we're going to allow it to interact with water, which is in biological systems, usually necessary unless the purpose is like a lipid to create a barrier between environments that are aqueous, between 2 different water environments, then we would not want the charge, right? But in this case, we do. So to add some functionality and allow this molecule to become water soluble, add here an oxygen. So now the oxygen has 6 electrons in its outer shell. It needs 2 more. And so one of the bonds is going to be here with a carbon. The other is then going to be also with a hydrogen. But this is unequal and this is unequal. These don't share equally. This does not share equally. So the oxygen ends up being negatively charged. The hydrogen ends up being positively charged. These are partial charges as well. And now this molecule can interact with water. And this now is ethanol, right? Or ethyl alcohol, which is water soluble. So that's our first functional group, which is the hydroxyl group. Usually it is typed out, like if you're typing it in to a document, OH is typically the way it's done with a little dash because showing that the bond between a carbon, say for example, the carbon's not part of this, just showing you, is to the oxygen, not to the hydrogen. Right? The hydrogen's only bound to the oxygen. But I'm not drawing the carbon because we typically don't draw those. I'm just letting you know that's what this little dash means. It's not a negative. It's not a charge. It just means that's where the bond would be. It's a hydroxyl group. Hydroxyl groups are polar, and that's their main characteristic. That's it, and they're very common. We see them in carbohydrates all the time. So remember one of the rules of a carbohydrate, which is something I do in another lecture. But again, remember all carbons of a carbohydrate, I'll just do a simple one here, are going to have an oxygen attached to them. Most of those are hydroxyl groups, OH, like this. But one of them is going to have a carbon-oxygen double bond. All right? And that's going to be our next functional group. Okay, our next functional group is called the carbonyl group. So it's a carbon with a double bond to an oxygen. So this actually would be a carbonyl group. And so in the structure of a carbohydrate, it could be for different sugars. It's at different locations. So it could end up being, here. This could be an interior one, or it could be exterior, like that. But there's no hydrogen associated with that oxygen. It's the two bonds that it needs to form to fill that outer shell with electrons. Both come from sharing with carbon. So this carbon wouldn't have any other hydrogens associated with it. If I filled out the structure, there would be no hydrogen there, hydrogens, no hydrogen there, hydrogen, hydrogen, hydrogen. Right, and this would be C4, H8O4, just a super generic, carbohydrate structure. So, but following rules, certain rules, which I set up in another, again, lecture, every carbon has an oxygen. One of those is a double bond, the rest are OH, they're hydroxyl groups. So the carbonyl and the hydroxyl group are both essential to sketching out the structure of a carbohydrate, all right, and understanding it. So and the purpose of those is to add polarity. So sugars, right, are very water soluble. Why are they water soluble? But because of the hydroxyl groups, because they have a lot of them. And the carbon-oxygen double bonds are also polar as well. They're partially charged, the negative charge, so that's going to allow interaction with water. All right, so we have our hydroxyl and our carbonyl group, both polar groups. Now what we're going to do is add in something else here. I'm going to just put this. We're going to create a group here called a carboxyl group. Carboxyl group is very important. It's also another polar group, and it's sort of a combination of these two functional groups. It's like a hydroxyl attached to a carbonyl, forming yet a new type of group that also takes on a new characteristic as well. So a carboxyl group is sometimes just typed out. So again, if you're typing this in on a keyboard, you can put the little dash to bond to a carbon. But then the rest of this is not structural. This is not how it is structured. It is not OOH like this, right? That is, again, the common way. If you read this in a text in your book somewhere, that's what you'll see representing a carboxyl group. But that's not exactly what it looks like. I'll show you here. So what does it look like? There's a carbon. That's this carbon. That's part of the functional group. It's not one of these carbons. It's another carbon. That's part of the carboxyl group. Then we're going to have a bond to an oxygen that's part of a hydroxyl group, OH, like this. And then we have a double bond to an oxygen, right, like the carbonyl group. So the carboxyl, so this is now not ethanol anymore. This is the carboxyl group. Right. Now, obviously, it's going to be polar because this is going to be more electronegative than the carbon, carrying the negative charge. This one's going to be more electronegative. This one's going to be positive, and so on. So that's where we start off, and that's what you need to know, and that's how we're going to represent the sketches of carboxyl groups when you see them in fatty acids, when you see them in the amino acids, and other places as well. That's the main representation of them. Now, what you also need to know is that there's another function to the carboxyl group. Carboxyl groups are weak acids. So we sometimes see it written as COO negative, like that. What does that mean? Well, what that means is that this oxygen is sharing electrons with a hydrogen, but it's unequal. There's a strong pull. But it typically doesn't ionize on its own, like this, just as the hydroxyl group. In this case, though, what we have is additional pull, right, from this oxygen here, pulling the electrons from this hydrogen, even though they're not connected, they're kind of indirectly connected here through the structure. And what happens then is that extra pull given to it by this extra oxygen here tends to actually steal the electron from that hydrogen, and this takes on a full negative charge. Okay, so it's ionized. And the hydrogen, which is actually a proton, goes into solution. The definition of an acid, remember? Acid, or pH, right? Our pH is a measure of hydrogen ion concentration. Acids donate hydrogen ions or protons to a solution, so this carboxyl group has just behaved as an acid and given up a hydrogen ion to solution. It's weak because it's very reversible, right? So that hydrogen, that proton can come back, and then it becomes the same group that we had here in the beginning, all right? So for my class, I prefer you just draw it in the non-ionized form most of the time, unless I've specifically asked for it. Otherwise, keep it that way. It's much easier to draw our other connections and bonds between different molecules when this is involved in the bonding, all right? But you need to know that that's a characteristic of this group, is that it behaves as an acid, and be able to kind of explain that, like why it does it or how it does it. So generally, again, I would say don't draw it in the ionized form. For me, just leave it in this non-ionized form. But know that it would be, polar, and that it could ionize. So now we're going to move on. We have hydroxyl, carbonyl, and then together they're kind of forming a carboxyl group, which is this guy. So now we're going to add something else. And I'm going to change this up a little bit because I'm going to show you again like the functional groups and how they form the biological molecule. So you know your functional groups, you'll know your biological molecules. It's almost, not that you don't have to study them, but if you study this, it makes studying them so much easier. So our next functional group is going to be the amino group. Amino group is written typically as NH2, like this. Now, an amino group can behave as a weak base. It's another characteristic. And as a weak base in an ionized form, we then write NH3+, so it actually has the charge because it's removed or taken up a hydrogen ion from solution. So let's look at an amino group. The amino group here is going to have a nitrogen forming a bond typically with a carbon. That's part of some core molecule. Nitrogen has 5 electrons in the outer shell. So one of those is going to form a pair and bond with this carbon. Another is going to pair up and bond with a hydrogen. And another with another hydrogen. Okay, so it's NH2. This right here. This is the amino group. That. Now we said it's going to be polar. So, sorry, wrong way. Nitrogen's more electronegative than hydrogen, so that's going to be negative. These will be positive, making it polar. It could behave as the base, so that means sometimes protons or hydrogen ions from solution might be attracted to this nitrogen, giving it a full charge, resulting in an actual ionized version. Like once again, I prefer just draw it in the non-ionized form. It'll make it easier for the other things that we're going to do in my class, but keep in mind that it does ionize and that it does behave then as a base when it ionizes, and it takes up protons. Now the reason I do it like this is also, and I left the carboxyl group up there, is because what you have in front of you now is an amino acid. Amino group, carboxyl, acid group, this is the amino acid. So if you going to draw an amino acid, which is important in my class, you need to be able to sketch out an amino acid and understand its structure. There's a core central carbon, and attached to that is an amino group on one side and a carboxyl group on the other side. So amino acids, first off, very polar molecules, all kind of partial charges, all over the place, right? From both of these functional groups. interesting and kind of odd thing, about them is they can behave as a weak acid and a weak base, kind of as both. When you talk about amino acids, one of the things that we do is we say these amino acids are nonpolar amino acids, or the polar amino acids, or the acidic amino acids. And that's just because when we say those things, what we're talking about is whatever is attached here for an amino acid as the R group. And so what that would mean is that if it's an acidic amino acid, there's an extra carboxyl group up here. There are 2 carboxyl groups. If it's a basic amino acid, there's an extra amino group, which should be up here. If it's a non-polar amino acid, then this part is just hydrogen or carbon and hydrogen, and no functional groups at all. So you see, so when we describe the amino acids, they all will have this structure here. 1 central carbon, 1 amino acid, 1 carboxyl group. And then what's variable about them is whatever's attached up there. And that could be a whole variety of things. All right, so we have hydroxyl, carbonyl, carboxyl, amino, and we can see how they start to come together to form our molecules, our carbohydrates, our amino acids, and so on. So this is why you study these. So as you study those other molecules, they just all fit together. And so it just makes a lot more sense, and it just makes it a lot more easier if you're going to sketch things out to be able to do it that way. And kind of think of it that way, and that way you'll know also if you make mistakes, because you'll know that, well, that does not look like an amino group on my amino acid. I must have done something wrong. So if you know these first, it helps. Last 2, one of them I'm going to go through really fast here, the sulfhydryl. Amino group. Sulfhydryl is very simple. All right, it's just sulfur, hydrogen. That's it. The sulfur is attached to a carbon, so you have some molecule. Hydrogens attached to a carbon here like this. And then you get a sulfur attached to the carbon. That sulfur then also bonds to a hydrogen. And that's it. It's polar, just like the hydroxyl group. The electronegativities of the sulfur and the oxygen are similar. If you look in the periodic table, they're stacked there in the same column. So it just behaves like that. This one's partially positive, partially negative. It just adds polarity. Now that's the main thing. We don't see these very often. They're not all that common, but in the study of protein structure, what you'll find is that there's an amino acid that has a sulfhydryl group as part of that R group. And what they can do is then bind specifically with other sulfhydryl groups to form a unique type of bond called a disulfide bridge. It's a covalent bond that's very important in stabilizing 3-dimensional structure of polypeptides or proteins. So that's kind of where it comes in. So what do you need to know about sulfhydryl group? Very simply, 1 sulfur, 1 hydrogen. That's it. It's polar, and it can add polarity. But in addition to that, it's important because it's involved in this special, unique bond that's part of stabilizing protein structure. And that's it. So this one's just kind of quick. Now, the last one is the most complex one, really. And not the most important, but it's a very important one. And you'll see why in a minute here. So this last one is the phosphate functional group. So phosphate functional group, again, typed out. What do we usually do? We would do P O4 -2. That's kind of the way we write it out. So what is it? It is I'm going to erase this. Phosphorus. It's going to have a bond to 4 oxygens. Okay, PO4, like this. Now, one of these oxygens is going to have the bond with a carbon. So, to a carbon. I'm not going to draw this out here. I'm just going to write it out so you can know. That's not a negative. That's just the bond, okay? Joining it to some other molecule. One of these is going to be a double bond. So there's 1 double bond. So there's 4 oxygens, but there's actually 5 bonds here around the phosphorus. Keep that in mind. These two other oxygens, these actually have negative charge. Okay, so these are ionized. So that's full charge. So full charge. Not partial charges. It's not from a polar covalent bond. That's an actual negative charge. I'll really kind of emphasize it here with another color. Okay. How does that happen? Where do those electrons come from? Sometimes people ask me that, and so the answer is, at one point this would be a phosphoric acid, which is a very strong acid, a powerful acid. And the difference between powerful and weak acids is that the strong acids, when they give up a hydrogen ion to solution and leave the electron behind, it's typically not reversible. It stays apart, or it doesn't reverse typically. So when that happens here, it just takes on the full charge. And whenever we see a phosphate, it's just always going to have the negative charge. So I told you about the carboxyl group. I said, don't necessarily draw it in the ionized form all the time. For me, draw it in the non-ionized form with polarity, but just know that it does that. And the same with the amino group. But for this one, you must sketch it in the ionized form, because that is the way it is just most commonly seen pretty much all the time in all the structures that we have in the cell. Now, where do you see the phosphate functional group? Well, it's very common as part of very important molecules like ATP. All right, so the P of ATP, so we have adenosine, which I'm not going to draw the whole structure of right now, and then you're going to have phosphates. So actually it would be an oxygen. Like this. Oh, like that. Oh. Negative, negative. Oh, I'm going to do this in different colors just so you can emphasize it. Okay. Negative, 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 negative. So the reason I'm drawing this out, so an ATP molecule, adenosine triphosphate, 3 phosphates. All right. Another thing to take note of is that negative charge, actual full-on negative charge. Okay, and there's also polarity as well, but it's negative. All right, and the same thing. Phosphate groups are part of nucleic acids, so our DNA and RNA nucleotides have phosphate groups attached to them as well. And it's very important in the connecting of the nucleotides when we build the DNA molecules as well. So being familiar with the structure of a phosphate is extremely important for a whole variety of molecules that are some of our extremely important molecules that we study. So make sure you know this group. So what do you need to know? Again, names, that they basically are all polar, how to do a simple sketch of each of them, how would you kind of write or type them out, and if they have any unique property, what is that property? And then also, which biological molecules might you find them in? So those are the types of things that you should be focused on when you're studying the functional groups.